Welcome, everyone. Good morning. So we are going to continue in our journey through the cell by now focusing today on eukaryotic cells. And a reminder that I asked you to do some homework. So if you were able to do it, please make sure that you've brought it with you to support you today in the class, your strip sort and your mitochondria and chloroplast drawings. Sort of an overview of where we're going. We're going to talk about cell walls today, plasma membranes, and flagella and cilia, ribosomes. A lot of this we've discussed already for the prokaryotes, so we're going to focus on the similarities and differences with prokaryotes. The endomembrane system, mitochondria and chloroplasts, which are uniquely eukaryotic, and then endosymbiotic theory, which nicely wraps it up and kind of talks about the possible evolutionary relationship between these two major cell types. I imagine that this picture is quite familiar to you in some way, shape, or form, right? This is sort of an iconic eukaryotic cell picture with an animal-like, perhaps protozoan on the right, and a plant-like, could be algae, could be plants, cell on the left, where we can see some of these common structures like the nucleus and some distinct ones like chloroplasts and vacuoles. To get us warmed up, and just to be very clear at the outset, this is not a question that would appear on a test, since this is not, I'm assuming you know this stuff already, so I'm sort of basing that, but I just want to warm us up and dig up some of those old memories. So I'm not going to show you a picture of a cell on a test and ask you to label it, but we will do that right now to kind of warm up and remind ourselves what are the components of the eukaryotic cell. And so I'm going to do a little cold calling here, just like I did last time with the bacterial cell, that you could see that there's more things to look at. And I want to remind you of the rules of the cold call, which is if you don't have an answer that you feel comfortable to share, you can always pass. And that is a perfectly legitimate and kind thing to do for yourself and other people. But with that said, if you have an answer or you just want to wildly guess, those are also welcome. All right, so we're going to start with A1. A1 is pointing to this thingy here that's kind of around in several different places. And we're going to start with Alyssa. Alyssa, what do you think A1 is pointing to? Or do you want to pass? That's also fine. Are you able to unmute, Alyssa?
I think I'm gonna pass. I thought it was a rough, or I thought it was the ER at first, but it's not. It might be the envelope.
Nuclear envelope is a great guess, and it is correct. Good job. Okay. Next one, we're going to go to A2. A2 is a little bit hard to see what it's pointing to. It's pointing to all these little holes. So A2, we're going to go to Stephanie.
Is it the nuclear pore?
Yes. All right. A3, we're going to go to Aminata. What do you think all these little squiggly bits are? Oh, there's some problem with Aminata's having some difficulty with her microphone. So yes, yes, Aminata, you can go. Let's instead ask Andrew. All these little squiggly bits. A3, what's A3, Andrew? What do you think?
Chromatin.
Yes, okay. Next, we're gonna ask Sarah for A4. What do you think A4 is?
Read the nuclei?
Yeah, the nucleolus. Yes, indeed, that's right. Okay. So next we're going to go to, so now the whole thing, this refers to the whole things. A is referring to the sort of collection of all these things. And so we're going to ask Stasia to tell us what A is.
The nucleus.
That's correct. All right, good. So for B is pointing to a number of things that don't all look identical, and they look like little strings or tubes. Nicole, what do you think that is, if you have a guess?
I think it's the cytoskeleton.
Yes, and that would be a good guess. All right, for C, C is pointing to all of this space inside of the cell, this fluid space, like from all the parts, like sort of are in here. So let's do that as a chat. What do you think all this space is called? Put that into the chat if you know. I'll call out some of you. Yeah, indeed. The fluid-filled interior of the cell is called the cytoplasm, just like it is in prokaryotes. Great. All right, D is pointing to this kind of orangey pancake-looking structure. Arian, do you have a guess or do you want to pass? Either is fine.
Can I pass?
You can, absolutely. So for D, again, this orange structure, let's ask Bimi, what would you say?
I think it's a smooth endoplasmic reticulum.
It's close. It is part of the same system, but you'll notice it has kind of this layered pancake looking sort of appearance and is more flattened than the smoothie are, though they are quite related. So that is a good guess. Braden, would you have another guess for what D might be?
Not really. I would have guessed the same thing.
Excellent. All right, I'm going to ask one more person, then I'm going to open it up for the class. I know I'm putting people on the hot seat, but I appreciate those of you who are saying, you know, I'm not really sure. I'm going to say it's this, and that's not right, but those are all wonderful answers. Okay, Melissa, do you have a guess for what D might be?
Is that the Golgi apparatus?
Yes, that is intended to depict the Golgi. The sort of stacked pancake looking appearance is sort of what it distinguishes it from the smoothie art, though that is, they're quite related, as we'll see today. All right, so E is next. E is this little pouch coming off of the Golgi, but notice this part, it says it's acidic. So it's a specific type of what this is. And we're gonna ask Mai to tell us what she thinks.
Oh, is it a lysosome?
It is a lysosome. Good job. Okay, next we're going to ask Charisse for F. F is a process where something is being dumped outside the cell. So what might we say about that? Charisse, any guesses?
Sorry, can I pass on that one?
You absolutely can. Yes, I'm going to ask Mabintu if she has a guess for what F might be, this process of dumping stuff outside the cell. Are you able to unmute? Go ahead.
So is it exocytosis?
That is correct. Good job. Exocytosis indeed. All right, G is up next. G, Courtney, would you have a guess for what G is?
Free ribosomes.
It is a free ribosome. Good job. Okay, next we'll go to Emily for H. What do you think H is? Your microphone doesn't work. Do you have a guess that you want to put in the chat? If you want to pass, that's also fine. Plasma membrane, yes. All right, now we have I. For I, I'm going to ask Lanisa.
Is that a mitochondria?
It is a mitochondrion. All right, J. Let's go to Jordan for J. Why not?
That's the smooth ER.
Yes, that is correct. All right, and Katie, do you have a guess for what K is? This little pouch here.
I'm not really sure. I think it may be a peroxisome, but I'm not sure.
Yeah, it could be. It could be. It is intended to show something more general, but peroxisome is not a bad answer, actually. I would accept that. Does anyone have another answer that also would make sense? Raise your hand if you do. Brooke, go ahead.
Maybe a vesicle?
Yes, it's intended to depict a vesicle. But again, some of these things are a little bit like, there's more than one answer that would make sense for what it's pointing to. So I would accept either of those, but yes, vesicle. Okay, good. L. L is pointing to what? Let us ask Erica Marston.
That's the rough ER.
Rough ER, yes, indeed. M, Erica Lowry, what would you say M is?
The I suck at saying this word. The phalangium.
Yeah, the flagella, yes.
Thank
Or
you.
flagellum. Flagellum is also the singular flagellum. That's also accurate. Good job, you guys. Okay. Now we have a couple more things to see. Now, first of all, we're in a different cell type. What cell type is this? Put that into the chat. Okay, so both of them are eukaryotic. This is a plant. Yes, this is a plant cell. And how do we know? Right into the chat, any of the items in here that are called out by these little numbered lines, what are we looking at that tells us, oh, it's a plant, not an animal or protozoan? So we see a cell wall. That's what animals and protozoa do not have. We see a chloroplast. This is uniquely found in plant cells or algae. And then there's this large vacuole called the central vacuole. Good. Okay, so again, if you want to review those, because there were some that were like feeling kind of iffy about, I would go review it on Canvas. There, that same picture is there with all of the answer keys for what the things are. You want to remind yourself what the function of those different organelles and so on, and what they're composed of, because we're not going to cover that in this class. I'm going to assume that you kind of know that. I'm going to lean a little bit on that in this class, but if you need to review them, that is there available for you. Sarah, do you have a question?
Yeah. So do you have to know them for the exam, like the structure function?
In a way you do. So let me sort of see if I can clarify what you need to know. I'm not going to ask you a question like, what is the function of the endoplasmic reticulum? I'm not going to do that. Or I'm not going to say, what is a vesicle for? But I might ask you a question, an application question, which assumes that you know what that organelle does. Like if I were to talk about lysosomes, I would assume that you know it's digestive. And so that's why you want to review and refresh your memory about those structures. We're going to focus on the endomembrane system today, and you'll see a little bit more about some additional information that you might not have been taught before that you have to know as well. Does that help?
Yeah, thank you.
Great. Okay, now this is not, you won't be presented with this on a test either, but I want to illustrate the powerful information provided by the electron microscope. And you can see with the electron microscope, all of these incredible, this is not a drawing, this is an actual image on an electron microscope. And so looking at this for the first time, if you had been using a light microscope up until now, you would see a lot of interesting, obviously functional, structural things that you don't know what they are yet. Now, with the benefit of your education, you have a better shot at labeling these things than the people who first looked at these pictures, even though they developed the technology to do so. So I don't want to do a cold call or put anyone on the spot for these, but if there's any one of these items that either you feel like, I think I know what that is, or I want to see if I know what that is, or if there's something you're particularly curious about, then raise your hand and let's just talk about it briefly before we move on. Anything in here there where you're like, I think this is this, and I'm not going to correct you or comment it on all. I'm going to show the answer key in a second, but I'm curious to know if there's any of these that you have a guess about. Raise your hand if you want to make a guess. Charisse, what would you like to guess? Tell us the letter and what is the item pointing to?
I'm guessing D is a chloroplast.
Great. Anyone else have a guess about anything they see in this picture or a question? Aminata, what are you seeing?
I'm guessing B is a nucleus.
Great. And Andrew, what about you?
I'm going to guess for B also, but I'm going to guess central vacuole.
Great. Anyone else have a guess or want to put forth or a question about any of these, or just point out something that looks really cool? They don't know what it is. Alina.
I think E is maybe Golgi.
Excellent. Okay. Anyone else? Anything else that's sort of looking particularly interesting to you? Erica.
I just had a question about B. If it's a nucleus, because I did think it was a central vacuole as well. So I was just wondering why it's structured like that if it is a nucleus.
Great question. And again, I'm not going to comment on any of these things until we show the answer key, but that is an excellent question. If you were looking at this with your colleagues, right, you would all, these are all legitimate comments that I imagine some of them were like, well, I wonder what that is, or is this, could this be this? And in fact, some of your guesses are much more sophisticated than they would have been able to make because they didn't even have a word for some of the things that we can see here. Anyone else? Anything else you want to comment on or ask about or point out or say, I think this is this? Let's take a moment to see if there's anything that you feel like you want to mention. Nicole.
I think J is the nucleolus.
Great. Okay. Anyone else? Anything else that's popping out to you? You're starting to feel like, ooh, I wonder if I want to cast my vote in. Charisse.
I'm going to say A is a peroxisome.
Great. Last call for, Bimmy says G is the plasma membrane. Excellent. Okay. Wonderful. And then, all right, so I think we're done with our guessing. Good job. And some of you who maybe weren't as brave, but did nonetheless have thoughts in your head, just keep that in mind. Here's the answer key. This is what all of the things are. Now that we have the benefit of many decades of study, okay, but seeing this for the first time be hard to know. So just have a look and see if anything's surprising to you. Just to address the earlier question, actually, I'm not entirely remembering what the earlier question was. So Erica, I think you asked a question. Could you restate it for me, please?
Yeah, so I was just wondering, because it's a nucleus, why is it shaped like that? Because that wouldn't have been my first guess.
Yeah, that's a really good question. Honestly, I don't know the answer to that. I think one of the other things that stands out to me is that, and honestly, me personally looking at this picture, I would have guessed that to be the vacuole. But it is large, and the nucleus tends to be large, and the animal nucleus is also large. I think the thing that sort of stands out to me as being quite different is that there's a lot of density in the chromatin, and there's a nucleolus, and that's not really apparent here in the plant, so that's confusing to me too. However, what I do notice is that this structure does have a double membrane, and then when I see that, I'm like, oh, well, that does make more sense. That could, but why is it shaped differently, and why does it have a different sort of density appearance? I'm not really sure. Those are really good questions. And what I will say in general about nuclei is they do not all look the same. Even in human cells, even in human white blood cells, if you compare, let's say, a lymphocyte, like a B or a T cell, to a neutrophil, which is a white blood cell that's a phagocyte. And if you look at the shape of the nucleus, it's very different. And so that's typical, actually. It's not always round, it's not always one piece. Some of the human cells have nuclei that have lobes, which is very weird to look at if you're used to this kind of stereotypical image. Great questions, guys. Excellent. Thank you for all of those contributions and thoughts and comments and questions. Let's talk about the cell wall in eukaryotic cells. Okay, so in prokaryotes, the function was protection from osmotic lysis. And in eukaryotic microbes, that's still the case. When it comes to macroscopic eukaryotes, there's more complexity, but in terms of microscopic eukaryotes, that's largely what it is for. Which eukaryotic cells have cell walls? Write that into the chat. Which eukaryotic cells have cell walls? Which types? Yes, plants do. And then in the microscopic world, algae and fungi. Yeah. And so notably, the eukaryotic animal cells and the eukaryotic protozoa, which are the animal-like single-celled organisms, they do not have a cell wall. Now, the composition of the cell wall in prokaryotes, in bacteria, it was peptidoglycan. In archaea, it was pseudomurine, which is not something you have to remember. But both of those are sugar and protein based. The composition of the cell wall in eukaryotic cells is what? protozoans are protists, yes. Yes, Andrew, they are. However, and it's important, so in the chat, I'm just responding to something Andrew said. Protists are a large group of single-celled eukaryotic microbes that include things that are animal-like and plant-like, and the animal-like ones do not have cell walls. Those are the protozoa. Yeah, so in the eukaryotic world, plants and algae have cellulose as their cell wall primarily, and fungi, including microscopic ones like yeast and mold, have chitin. Now, these two are very similar in the sense that they're made of polymers of glucose, which is different than the bacterial cell wall altogether. The cell wall of the bacteria is made of peptidoglycan, which has sugar, but not glucose. It is a sugar called N-acetyl muramic acid and NAG and NAM, if you remember that from the chapter 4 text and the notes. And it usually has proteins attached to it as well. So this is a little bit different. Bearing that in mind, and thinking about this question I asked you last time, penicillin being this miracle drug that's antibacterial but not harmful to humans, which was an innovation, a major step forward from previous treatments that were toxic to basically all life forms. And we established that it was so miraculous because it specifically targets something bacteria have that we don't. And what was that target? What was it penicillin specifically targeting? Yes, and we can get as specific as necessary. So it specifically targets the cell wall of bacteria. It specifically targets that because it's attacking peptidoglycan, which is in the cell wall of bacteria, which we don't have. And it specifically attacks the cross-links in peptidoglycan of gram-positives only, so not gram-negatives. Now, bearing that sort of specificity in mind, let me ask you a question that once you take it into your heart will become obvious, but frequently for people is not obvious when they first step into the classroom. Let's suppose you had a toe fungus and you needed treatment. You needed some kind of drug to treat your toe fungus. Would you take penicillin? All right, everyone is in agreement that this would not be an effective treatment. Raise your hand and explain why that's the case. Why wouldn't you take penicillin for this toe fungus? Andrew, go ahead.
You wouldn't take penicillin for toe fungus because the group fungi doesn't have peptidoglycan or a cell wall made out of peptidoglycan.
That's correct. The specific molecular target of penicillin is only found in gram-positive bacteria. It's not found in fungi. They do have a cell wall, that's true. And if all you remembered was that penicillin attacked cell walls, then you might be tempted to think it could attack fungus, but it can't because fungal cell walls are made of something different. Okay. So let's talk next about another structure that is somewhat similar between prokaryotes and eukaryotes, but has nonetheless important differences. So flagella and cilia are shown here, and they're discussed together because what they're made of is the same, okay? So while they also share functional similarities. So these are eukaryotic structures, eukaryotic cells, and you can kind of see that on this scale bar, right? This is enormous compared to a typical bacterium. Hopefully that's obvious. So the function of these two structures is similar to, what is it? Put that into the chat. What are they for in the microscopic world? Now, when we get to the macroscopic organisms, it gets more complicated. But in terms of the microbes that have them, the eukaryotic microbes, it is for motility, correct. And whether you have this one long one or a whole bunch of little shorties all over the place, they're both made of the same thing. And what is that? Correct. They're made of microtubules. And in fact, it's not just sufficient to say microtubules. You want to understand that it is what we call a 9+2 array, which is very jargony, I know, but it's a 9+2 array of microtubules. It's a whole bunch of microtubules organized in a very particular way. 9 pairs in a circle with 2 in the middle. if we remember what our prokaryotic flagella was made of, it was made of what? Just remind us what the prokaryotic flagellum was made of. Yeah, it was made of a protein called flagellin. And what's the width of a prokaryotic flagella? What's the width of a prokaryotic one? Right, 15 nanometers. Now, I want to tell you that each microtubule is 25 nanometers wide. So the bacterial flagella would fit down one of these little straws or tubes, just one. So we're talking about something that's substantially larger. You know, it looks the same in a way, it has the same function and it kind of looks the same, but that's only because of scale. That's only because this cell is quite large that this looks the same. But remember that bacteria are tiny in comparison to this, right? The little bacteria would probably be just a little tiny tip of this guy. And so its flagella is going to be much, much thinner than this. Pardon me. So what I'm saying is that this structure is more than 10, more than 15, maybe more than 20 times thicker than a typical bacterial flagellum. And consequently, you can see it very faintly on a light microscope. It is big enough to be seen with the right optics. Now, when we describe its motion, there was a way we described the motion of the bacterial flagellum. How would we describe this one when we look at it moving? What's the best descriptor? Yeah, the eukaryotic flagella has a wave-like or whip-like motion. And if you've ever seen like the iconic video of a sperm, a human sperm wiggling, then that's the image you want to keep in mind. And that's distinctly different than bacteria because how did their flagella move? They rotate. So if this was a bacterium, there'd be a motor right here at the base where the flagella attaches, and that's the only part that moves. It spins and the whole flagella is just a limp noodle that spins around because of this motor. But in a eukaryotic flagella or cilia, the motor proteins are all up and down here, and they're moving the tubes with respect to each other, with respect to the central part as well. So the entire thing kind of writhes or has motion. It's not just due to a motor at one end. All right, now in prokaryotes, there were glycocalyx. There were two major types. What were those two major types? Write that into the chat. What were the two major types of glycocalyx? Yes. Capsule and slime layer, that's correct. Now, what was it made of? What's the composition of the glycocalyx? That's right, it's made of carbohydrates, a complex carbohydrate network. And sugars, as you may know, at the molecular level, attract water, so it tends to be like a little bit like a jelly or a slime. Do eukaryotic cells have a glycocalyx? Some of them do. Yeah, just like prokaryotes, it's not every cell, but some of them do. And in the case of, say, human tissue, it's often part of the first line of contact with pathogens. So environmentally facing tissues like your mucous membranes, those cells have a glycocalyx that sort of underlies the mucus layer. All right, when we go to the cell membrane, we remember that just like in prokaryotic cells, the major function of the membrane is the same. And what is that function? Exchange, correct. Exchange and communication. And the structure is also similar. So what is structurally similar between these two, between all membranes really? It's a phospholipid bilayer, and there's membrane proteins embedded in it. That's right. There are, however, functional differences. One of the things we mentioned last time is that bacteria that can perform respiration or photosynthesis put the machines that they need to do that into the plasma membrane. And this is not true in the eukaryotes. The eukaryotes put those machines in mitochondria and chloroplasts. There's another functional difference. What is something that the eukaryotic cell can do that the prokaryotic cell cannot do with its membrane? Some of you have already mentioned it. Good job. Yeah. It's a process of importing stuff from the environment in bulk, basically, known as endocytosis. Now, a really important thing to understand about this and why this endocytic process is important for eukaryotic cells is in a bacterium, if you want to bring something in from the environment, that's a great question, Andrew, and I'll address that shortly. If you want to bring something in from the environment in a bacterium, then you've got to import small molecules one at a time. Okay, so let's say you want to bring in a sugar molecule and you need a glucose transporter. You can bring in one sugar molecule. You can't bring in a whole like polysaccharide. You can't bring in a whole protein. You have to go one amino acid at a time. So endocytosis allows you to bring in 100 times or 1,000 times that all at once. Think about phagocyte in the human body. They can engulf an entire bacterium, which is massive, so it's quite a big deal. Now, why is that helpful or important? Remember that eukaryotes are much, much bigger, and so they require faster exchange, and this is one way to achieve that. So it turns out that yes, there is some exocytosis in bacteria, and we don't discuss that in a great deal of detail, but exocytosis is possible in prokaryotic cells. Okay, now there's also structural differences, primarily the fact that in eukaryotes, in addition to phospholipids and protein, there's also steroids in the membrane. Okay, so in animals, there'd be cholesterol, in fungi, there's ergosterol, and in plants, there's phytosterols. Okay, we now come to the cytoskeleton. Did prokaryotes have one of those, yes or no? Correct, they do not. What's the function of the cytoskeleton in a eukaryotic cell? There's more than one, and that's partly because the cytoskeleton is actually 3 different networks, so they all have different things they do. Yes, it can help preserve the large and fine shapes of the cells. It allows movement using cilia and the flagella or crawling movements. It provides internal organization and also internal movement. Organizing organelles, yes. And promotes cell division. So the shape of the cell, sort of the overall shape, is provided by microtubules often, and then finer things like little finger-like projections are supported by actin. Movement is also supported by both microtubules and actin, in the sense that flagella and cilia are are made of microtubules, and then if you have something like an amoeba that has crawling movement, they use actin for that. The interior of the cell is organized using microtubules, and also things can move around inside the cell in a very organized way on highways made of microtubules. And microtubules also promote cell division. Some of you also mentioned anchoring or support. So intermediate filaments provide strength, support, integrity, and the ability to create large tissues in some eukaryotic cells. All of these components of the cytoskeleton with all these myriad of diverse functions are made of what? What is the chemical composition of the cytoskeleton? Yes, that's right. They are made of protein, correct. And more specifically, they're made of microfilaments are made of actin protein, microtubules are made of tubulin protein, and then intermediate filaments are made of other things. This is kind of a summary of the three types and kind of what they do and how they look, how big they are. I'm going to give you just a little bit of a review quiz, if you will. One of the things that's really striking and cool about the cytoskeleton is, sure, those filaments, they're too tiny to see with a light microscope, like impossible. They're so skinny. The biggest one is 10 times too small to be seen. But what you can do is you can light them up. You can attach fluorescent dyes or fluorescent proteins to the filaments and specifically light up one of the networks, and as opposed to any of the others. You can light up actin only, or tubulin only, or keratin only. That will allow you to see where that is located. And you can look at it under an a UV microscope to see it. So up here is a picture of one of these. And this type is found kind of in these perpendicular bundles at the edges of the plasma membrane and facilitates crawling movements. What is it and how big is it in width? Yes, you guys in the chat are correct. It is a microfilament. It is about 7 to 8 nanometers wide. And then Charisse, was that you were going to give that answer?
Yes, sorry.
Perfect. Well, then you're right. And then Andrew, your hand is up. Did you have a further question? Or was that from before? Oh, great. Okay, just checking in. Yes, that is the actin. Okay. Next, we have this one. It grows out from a central organizing center, kind of like hair coming out someone's head, and it moves all, so it forms a network all throughout the cell. And it's hollow and it's rather large. And what is it? Yes, microtubules, and they're 25 nanometers wide. And last, we see the intermediate filaments that tend to form this network around the nucleus, sort of perinuclear and into the cytoplasm, providing strength and connection and kind of being an intermediate size. Yeah, great. All right, moving on to ribosomes. Now remember the prokaryotic ribosome, its function was protein synthesis. Its structure was 2 subunits, the 30S and the 50S. And together, when you put them together, they make the so-called 70S ribosome. This is a density unit. Are eukaryotic ribosomes the same? Let's start with their function. Is their function the same? Correct. They are still required for protein synthesis. Now, structurally, are there any differences? If yes, call them out. What might be different about the structure? Are they still made of the same things? Do they still have the same do they still have 2 subunits? Are those subunits the same density? What's the same? What's different? Great. Good. Okay, so the eukaryotic ribosome is similar in function and in terms of having 2 subunits that are made of RNA and protein, but you'll notice the density of those subunits is bigger. Okay, they're 40S and 60S instead of 30 and 50, and the total ribosome is denser as well. So you can think about this as it being bigger, like that would be helpful, but it's really a density difference. Another difference is that all of the ribosomes in the prokaryote are found in the cytoplasm, because that's the only place they can be. But in the eukaryotic cell, they're found in the cytoplasm and sometimes also attached to the rough ER. Are there 70S ribosomes in eukaryotes? Yes or no? there can be, and there frequently are. Raise your hand if you could explain why that's the case. Where are they in the cell? Raise your hand. Stephanie, go ahead.
They're found in the chloroplasts and mitochondria.
Yes. Importantly, and we'll touch back on this later a couple of times, chloroplasts and mitochondria are two eukaryotic organelles that have their own ribosomes. They also have their own DNA. And when you look at it, it's surprising because the ribosomes don't look eukaryotic. They look prokaryotic. They have this lower density. And the DNA also looks prokaryotic. It's a single circular DNA. Okay, we're not going to take our break yet. We're going to forge forward. We're going to quickly name every organelle we can think of. So you don't have to name all of them. Just put one into the chat and I'll just read them out. See if we can, again, just part of warming up, stretching our eukaryotic muscles or our eukaryotic ligaments and tendons, trying to get limber here. Name a eukaryotic organelle, throw that into the chat. The nucleus, the mitochondria, the endoplasmic reticulum, the lysosome, the Golgi, the chloroplast, the central vacuole, peroxisome, vesicles, smooth ER, lysosomes, yes. Okay, now there's a few that were mentioned that I'm going to call out, not to embarrass anybody. That's not my purpose. But there's a few that are actually not organelles, though they are eukaryotic structures that are only found in eukaryotes. But nonetheless, it's important to recognize they're not considered organelles. And that has to do with the definition of organelle, a membrane-bound structure, a membrane-bound structure. So things like the actin, microtubules, and intermediate filaments, that cytoskeleton, it's not surrounded by membrane. It's not considered an organelle. It is a eukaryotic structure for sure. Centrosomes, centrioles, and ribosomes are also not organelles. So then that's because they're not surrounded by membrane. Now here is a list of some of them, not all of them, just because we're going to focus on some of these. Okay, so we're going to focus on these all together. All of these together work together on several different goals. And so we're going to talk about them as a system, the endomembrane system. Also, mitochondria and chloroplasts, we're going to talk about their structure and briefly for two reasons. One, to support our understanding later of metabolism, because we're going to talk about respiration and photosynthesis, and it's really helpful with these complex processes to visually be able to map where that's happening. So we're going to talk about these briefly. And also the structure here will help us to understand the last topic of the day, which is the endosymbiotic theory. And we're not going to really focus on the vacuole, though, just we're going to acknowledge that it is an important component. And it's an interesting one because it's got a lot of different functions depending what cell type you're in the microscopic world, for sure. In algae, it provides storage and structural support, just like it does in plants. But in protozoa, it has a function that isn't really common to macroscopic cells, okay, like animals, like large scale animals. So it's used to provide water balance to some protozoa to prevent them from exploding in osmotic pressure and watery situations. And that's all I'm going to say about that. So we're going to focus on the endomembrane system first, and then these energy generating organelles second. So here's the endomembrane system. We want to remind ourselves like what is happening in each of these different organelles so that we can better understand how they work together to achieve certain common ends. The nucleus, of course, is where the DNA is found. And a lot of what's happening in the nucleus is that the genes in the DNA are being transcribed to mRNA. Now, the rough ER makes protein and smooth ER makes lipid. And so obviously the rough ER is going to depend on transcripts coming from the nucleus. There are vesicles moving stuff around all over the place, but one of the things that they do is they move proteins and lipids made in the ER to the Golgi. So there's another connection there. What is the Golgi for? Well, one of its jobs is to modify things chemically. especially stuff that's going to leave the cell. It decorates them with sugar, a process known as glycosylation. There are vesicles that bring protein and lipids from the Golgi to the plasma membrane. So vesicles are also doing that, like we've decorated with sugar, let's take it to the membrane kind of service. And last, there's a specialized type of vesicle known as a lysosome that comes off of the Golgi but does not go to the plasma membrane. Its contents become acidified and it becomes a lysosome. So I hope already you're starting to see the connections between these different organelles and kind of also like making a note of the way that I've described the function of these organelles, which might be slightly different than how it's been described to you before. So here is what the endomembrane system does. All those things working together have 4 objectives. They make all the exported proteins for the cell. So if the cell needs to send something out, let's suppose it's a B cell, okay? What is the primary job of a B cell? What does it spend its time doing? Making antibodies, and antibodies are proteins. So it's exporting proteins. So the endomembrane system is how that happens, okay? Also, cells have membrane proteins, and eukaryotic cells have lots of membrane proteins. They also have membrane lipids. So the endomembrane system makes these items and is how they arrive at the membrane. It's all of those organelles working together are required for that to happen. The endomembrane system makes lysosomes. It's also fair to say that lysosomes are considered to be part of the endomembrane system, but they are made by the endomembrane system, or perhaps one could also say the enzymes within them are made by the endomembrane system and then packaged into lysosomes. And last but not least, there are enzymes that reside in the ER and the Golgi, and they have to get there somehow. So they're made by the endomembrane system. So I'm going to ask you this question. Does the endomembrane system, specifically the rough ER, which we know makes protein, we've talked about how it makes exported protein and membrane protein. Does the endomembrane system make also cytoplasmic proteins, ones that are going to stay inside the cell, such as perhaps enzymes that are going to do important metabolic processes like glycolysis in the cytoplasm? Does the endomembrane system make cytoplasmic proteins? Yes or no? Just going to wait for everyone to vote. And it's fine for you to guess. I love guessing. Guessing is the best, right? If you actually don't know, saying out loud that you don't know is a brain trick so that when you get the answer, your brain will remember it better. So you always want to guess, even if you have no clue, it's fine. Great. So we have, some people say yes, some people say no, and that's pretty typical. Actually, the majority of my section last night said yes. And I will tell you before I reveal the answer, which is probably obvious from my tone what it is, but for several years, I thought the answer was yes. I have some part of my bachelor's degree, I don't remember exactly where. I've been studying biology for a bit, but I hadn't really focused on cell biology because, it's a big field. And I just thought the endomembrane system is how all the proteins are made in the cell. I didn't really know that that actually isn't the case. It's specifically exported proteins and membrane proteins, lysosomal enzymes, resident ER and Golgi enzymes that are made by the endomembrane system. But stuff that's going to live inside the cytoplasm of the cell and just like tubulin and actin, glycolysis, or things that are going to be in the nucleus, like RNA polymerase, DNA polymerase, those enzymes are not made by the endomembrane system. So we're going to focus on how this works together to make these things happen, but acknowledge that it's not the only game in town. Okay, so I am going to allow us to take our break now since we're kind of due. And when we come back, I'm going to sort of talk through this process, and then we're going to do the endomembrane strip sort activity. So make sure you have it close to hand. And I will see you guys at the top of the hour in 9 minutes. Okay, welcome back, everyone. So I'm going to briefly walk through the steps in the like how these members of the endomembrane system are connected for one example of the goals that they can accomplish together. And I want to start by just saying, sort of contrasting that with how protein is made for the cytoplasm. So let's suppose here's the cell, and let's suppose we wanted to make some actin, right? That's a protein that's going to live in the cytoplasm of the cell. It's going to be inside the cell forever. It's not going to go outside. It's just going to live inside and have its function there. So if you want to make some actin, then you go to the gene in the DNA for actin and you copy it into mRNA. And that mRNA leaves through the nuclear pore. It's found by a ribosome in the cytoplasm. The ribosome makes the actin, and now the actin is there. It's pretty simple. But if you want a protein that is going to end up outside the cell, or is going to end up in the membrane of the cell, you have to use the endomembrane system. And so the beginning is very similar. We start with whatever we're going to say. Let's suppose we're a B cell and we want to make an antibody for export. So first we transcribe the antibody genes into protein or into RNA, and that RNA leaves the nuclear pore. It's found by a ribosome in the cytoplasm. It translates it into antibody, but it does so by first docking at the rough ER and making the protein and inserting it into the lumen of the ER. So that's this fluid-filled compartment inside the ER. And then once that's been made, the antibody protein is packaged up into a vesicle, and that vesicle walks on the microtubule highway up to the Golgi and delivers its contents into the interior of the Golgi, so that antibody is now inside the Golgi. Because the antibody is destined to leave the cell, then the Golgi enzymes that live inside of here add sugars to it. And when that's finished, they package this antibody up into another vesicle that's destined for the plasma membrane. And when that vesicle reaches the plasma membrane, it dumps its contents outside, and that would be all the antibodies going out. So just to recap, if we want to export a protein, we first have to make it an mRNA, and then we do translate it with a ribosome into protein, but we insert that protein into the interior of the endoplasmic reticulum. Once that's finished, then that protein is packaged up into a vesicle that goes to the Golgi, and once it fuses with the Golgi, it deposits itself into the lumen of the Golgi, where it's glycosylated, packaged up into yet another vesicle, and sent to the cell membrane where it's exocytosed. Briefly, we're going to just review which process makes which types of things that the cell needs, which proteins are made where. So does the endomembrane system need to get involved, or is it sufficient for a cytoplasmic ribosome to do this for each of the following? And I would encourage you to participate even if your answer is a question mark. So imagine you're a B cell, you need to make some antibodies. Are those antibodies going to be made via the endomembrane system, or are they going to be made in the cytoplasm only? Great. Yes, they need to go through the endomembrane system if they're going to be sent outside. Okay, now imagine you're a T cell. The T cell has many membrane proteins, including the members of the CD4 receptor. You might have heard of this because T helper cells are decimated by HIV, and they have CD4 on the surface, so sometimes they're called CD4 positive cells. If we want to make membrane proteins like this, do we have to go through the endomembrane system, or is it sufficient to have a cytoplasmic ribosome do the job? That's correct. Endomembrane system again. Okay, one of the things that happens in the endomembrane system is that things that are going to go outside the cell or that are going to be displayed on the surface of the cell, like antibodies or receptors, they need to get decorated with sugars. And that process is known as glycosylation. The enzymes that do this job live inside the Golgi. They're called resident Golgi enzymes. If we want to make one of those, do we go through the endomembrane system or just make it in the cytoplasm? Right, we have to go through the endomembrane system. And last but not least, if there is an enzyme or something like a structural protein that's going to reside and function in the cytoplasm, do we have to make it in the endomembrane system or do we make it with a cytoplasmic ribosome? Great, that's right. Okay, now in the strip sort activity, I provided you with 6 things that happen, 6 events, if you will, that can happen in the various components of the endomembrane system. But they don't all happen all at once for everything, okay? So there's different things that these organelles work together to accomplish. And I asked you just to figure out what one of them was. If you're a B cell and you're making antibodies for export, what steps in what order do you need? Okay, so just figure out which of these events has to happen to make antibodies for export in what sequence, and then put those numbers into the chat when you are ready. Okay, so yes, first we have to take the mRNA, the nucleus generated, and translate it into protein, but that protein has to end up inside the interior of the ER. So that when it's packaged into a vesicle and goes to the Golgi, it ends up in the interior of the Golgi, where it is glycosylated because it's going outside the cell. And finally, it's repackaged into another vesicle and sent to the plasma membrane because it's inside the vesicle. When it reaches the plasma membrane, it gets dumped outside. So I'm going to make some breakouts for you to talk about some of these other things that the endomembrane system does. So we're going to work in groups of 3 or 4 to run the numbers for making a membrane protein. What would that look like? I'm going to create your breakout rooms. Make sure you have the strip sort activity. Maybe take a second to do a screenshot if you need to. But I want you to take a second to think about what your numbers might be. Okay, so write them down maybe. Don't put them in the chat, but write them down. So that when I send you your breakout rooms, you can compare notes. Now, once your group has decided that you all agree, then come back. And if you don't all agree, see if you can talk to each other about how you'd figure this out. But once you all agree, then come back to the main room. And that's how I'll keep a tabs on how much time you all need. All right, so as you're coming back, welcome back everyone. As you're coming back, go ahead and put your answer into the chat if you have not done so already. As you're trying to make membrane proteins, what are the steps you need to follow? Great. Okay. So this is the answer. Let's walk through it. A lot of you got there. Some of you were wondering if it was this and feeling a little unsure. But it's a very similar process to making exported proteins with one very key difference at the very beginning that makes all of the difference. So I'm going to say it in words and I'm going to go back and hopefully illustrate it in the pictures. The difference is that you start with step 2 instead of step 1. And the only difference here is in step 1, you make the protein and it's fully inside the ER. But for step 2, if you want to make a membrane protein, you have to start by putting the protein into a membrane right away. And so when you make the protein that's going to go to the membrane, the ER kind of knows, or the ribosome, I would say the ribosome knows that this is going to be a membrane protein. So when it makes it, it kind of shoves it into the ER membrane as it's being made. As a consequence, when this is packaged into a vesicle, the vesicle is made from the ER membrane. Okay, so the ER membrane is actually used to make the vesicle. So the protein's embedded in the vesicle membrane now. And when that vesicle goes to the Golgi and it fuses to the Golgi, the vesicle membrane becomes part of the Golgi membrane. So now this protein is in the Golgi membrane. And part of it is dangling out on either side. Okay, it's like a French fry kind of, or it's like a straw poking through either side of a screen, if you want to think about it that way. Part that's sticking inside is the part that needs to be glycosylated because eventually that's going to be on the outside of the cell. So that's glycosylation still happens. Then when you package it up to make a vesicle that's going to the membrane, you actually use the Golgi membrane to do it. So when you do that, you're taking a piece of the Golgi membrane, which has this protein embedded in it. So now it's in the membrane of the vesicle that's going to the plasma membrane. And when it fuses with the plasma membrane, it becomes part of the plasma membrane and the protein's already there in the membrane. Okay, so if we go back, if I can go back to the pictures, kind of, if we want to go to the membrane, so when the protein is made, instead of being inside the ER, it's actually in the membrane of the ER, kind of sticking out on either side. This membrane is used to make the vesicle that's going to the Golgi. So now the protein is in the membrane of this vesicle. When it fuses with the Golgi, it becomes part of the Golgi membrane. Now the protein's in the Golgi membrane, and the part that's sticking on the inside where the glycosylation enzymes are will be decorated with sugar. Then the Golgi membrane is packaged into a vesicle and it's now in the membrane of the vesicle. When it ends up at the membrane and this vesicle fuses with the plasma membrane, the vesicle becomes part of the membrane and the protein is already embedded in it. Andrew, that's a great question. We'll talk about that briefly in a second, though it's not strictly speaking part of what you have to know for this class, but it's a great question. And it's not really necessarily a simple answer, actually. So let's just go back through. My slides are kind of frozen. I'm just having to wait for my computer to kind of come back. There we go. So there's lots of reasons to decorate things with sugar that are leaving the cell. But the simplest way to say it is that it's functional. First of all, the surface of cells are covered in complex carbohydrates that have lots of purposes. There's lots of reasons to do that. One of which, in a multi-celled system, for instance, like ourselves, is for the purposes of providing the identity of the cell. That helps to sort of act like a molecular signature or a molecular name tag. But also sugars can provide part of the function of proteins, and proteins that are outside in the outside world typically have sugars on them as part of their structure that helps them to do their job. So that's the simplest way to say it. It's not something you need to know for this class, but it's an excellent question. And if you love the idea of glycosylation, you can read more about it. And if you email me, I can send you more like information in papers. Oopsies, I just revealed the answer, but maybe nobody noticed, or maybe you did and it's in your unconscious, or maybe you're a super fast reader and now you have it. But nonetheless, I'm going to send you away to briefly discuss another cell. And before I do that, I just want to, so I'll explain what the prompt is, but then I want to call something out. So phagocytes making lysosomes. Phagocytes are white blood cells in your immune system that gobble stuff up. And they do sort of a large endocytosis, right? They gobble up a whole bacterium, or a whole virus particle, or a whole protein, toxin, whatever. Thanks, John. But obviously, once they've gobbled it up, the purpose is to destroy it. So they use lysosomes, the digestive organelle, to do that. And so consequently, they need a lot of lysosomes. So if we were a phagocyte and we had to make some lysosomes, now lysosomes actually come directly off the Golgi. The lysosomal enzymes, the mRNA has to be made into protein, and it goes through the endomembrane system, ends up in the Golgi. The Golgi recognizes that this is a lysosomal enzyme and is not going to the membrane. It's not going to be exported. So it gathers them all up and packages it into a specialized vesicle. So what would the steps be? I'm going to send you off to your breakouts. When you're done chatting with your people and figuring it out, come back to the main room, and it should be relatively quick. All right. Okay. So as you're coming back, go ahead and put your answer into the chat as much as you were able to get to in your group. What your consensus was, if you reached consensus, or even if you just reached some part of it, weren't completely finished, but felt like this is as far as I can get. Great. Okay. So most of you got the same answer as me or pretty close. So let's talk about it briefly. If we want to make lysosomes, there's a couple things that are going to happen that are the same and just a couple differences too. First of all, the lysosomal enzymes are completely inside of the lysosome, so we don't need to stick anything in a membrane. We just put it inside the ER lumen, put it in a vesicle, send it off to the Golgi. And then the Golgi sees, oh, you're a lysosomal enzyme. I don't need to glycosylate you. I'm going to put you into the lysosome, and then it's done. Now, last but not least, what about those glycosylation enzymes that have to be inside the Golgi? What steps would be required to get them there? Put that into the chat. So I'm not sending you to breakouts for this one. You're just going to put them into the chat, see if we can all kind of get there. Great. All right, so the simplest way to do this is just, again, to make the protein in the ER lumen and ship it off to the Golgi. And now it's inside the Golgi and you're done. Okay. I do want to acknowledge a couple of things here. Maybe it's obvious, and certainly it will be obvious to any of you who have actually deep cell biology background, which is not the majority of you, but I am oversimplifying some things for the purpose of making it possible to understand it. You know, there's exceptions to these rules. It's also true that glycosylation happens to cytoplasmic proteins too, but that's via a different process. It doesn't happen in the endomembrane system. And also, you know, some lysosomal enzymes might actually be in membranes, et cetera. But this is kind of a nice, straightforward way of describing the majority of how this works. Like most of the time, this is how it works. If you want to export proteins, this is how you do it. If you want membrane proteins, this is how you do it, so on. Okay, now before we move on, does anyone have any questions about that? We do have to talk now about mitochondria and chloroplasts and about the endosymbiotic theory and get as far as we can towards being prepared for what's coming up next week. Great. So we next have to talk about mitochondria and chloroplasts. And again, I want to remind you why we're talking about them and focusing on them. There's two reasons to cover them now. They do important metabolic processes, which we will revisit in metabolism, notably respiration, cellular respiration. It primarily happens in the mitochondria, and photosynthesis, which happens in the chloroplast in eukaryotic cells. These processes can also happen in bacteria, and they're complicated, multi-step processes, and they're abstract, hard to kind of wrap your head around, you know, unless you just memorize them and then just sit with them for a few years. So it's very helpful for some people to have a physical structure to map where this stuff is happening, because it's easier for us to remember processes that we can diagram. So I want us to remember what the parts are of these so we can do that. And the other thing is that understanding their structure will partly help to support our understanding of the final topic today, which is the endosymbiotic theory. So just reminding ourselves what mitochondria look like. Okay, they've got two membranes, an outer membrane and an inner membrane. That creates two fluid-filled compartments that are separate, entirely separate from each other. the interior, which is the matrix, and then the space in between the two membranes, which is called the intermembrane space. So when you are looking at your diagrams, make sure that you have a sense of where's the inner membrane, where's the outer membrane, where's the matrix, that internal fluid, where's the intermembrane space, feeling comfortable knowing that it is in between these two membranes and completely separate from the matrix. What pH is the matrix? Bearing in mind that, you know, it's the site of the Krebs cycle. A lot of different biochemical activities are happening there. So lots of enzymes are working in the matrix. The pH is around a pH of 8. And it's, if you were to say neutral, I would also accept that. So it's neutral-ish. It's slightly on the alkaline side of neutral. And that's a good pH for the enzymes that do the Krebs cycle to function. Now, electron transport chain machinery is important for respiration. It's in the mitochondrion. Where is it specifically located? Yes, inner mitochondrial membrane. Whoops, inner mitochondrial membrane. Now, in the process of doing respiration, we have to create a proton gradient. So protons are actively transported from the matrix into the intermembrane space, and that causes them to build up in high concentration. And then they're allowed to diffuse back through ATP synthase across this membrane back into the matrix. That's what generates the ATP energy. Chloroplasts have actually 3 sets of membranes. They have outer, inner, and then this additional thylakoid membrane, which is like little pouches. And there are still 2 important fluid-filled compartments that are separate from each other, although the intermembrane space is not relevant to the chloroplast in terms of the function of anything important. But there's fluid that bathes the thylakoids, that's called the stroma. And then the interior of the thylakoid is also filled with fluid, and that's called the interior of the thylakoid or the thylakoid lumen. So you want to be able to sort of visualize a chloroplast that has these 2 membranes wrapped around these stacked thylakoid membranes, and the stroma, which is sort of exterior to the thylakoid, and then the interior of the thylakoid. Now, what pH is the stroma? Bearing in mind that this is where a lot of other important biochemical reactions happen, such as the Calvin cycle. So this stroma is basically functionally equivalent to the matrix. It's a site of a lot of biochemistry. The pH is on the alkaline side of neutral. So if you were to say neutral, I accept that, but it's actually closer to 8. There are electron transport chains in photosystems, which are membrane embedded, that are required for photosynthesis. Where are they located in the chloroplast? That's right. They're in the thylakoid membrane. Again, for this to work, we do have to build up a proton gradient, and we do it by actively transporting protons from the stroma into the interior of the thylakoid. So they build up inside these little pouches, and then they're allowed to diffuse back through ATP synthase, which is also on the surface of these pouches. And then the protons get back into the stroma and generate energy as they return. Okay, so we're not going to go through this, but you do want to be able to do this kind of comparative analysis. You can sort of looking at in mitochondria and chloroplasts, where are all these things occurring? My next question to you that's important though is, can prokaryotic cells perform cellular respiration or photosynthesis? Bearing in mind that in eukaryotes, this is done by the mitochondrion, And in eukaryotes, this is done by the chloroplast. Those organelles are missing in prokaryotes. Can they still do these metabolic processes? Yes or no? The answer is yes, as I've mentioned several times, so hopefully I've bashed it into your heads lots. So the question I have for you is, if the electron transport chains in respiration and photosynthesis have to be in a membrane, and those membranes that they're in a eukaryotic cell, like the intermitochondrial membrane and thylakoid membrane, are not found in prokaryotes, what membrane do they use? Where do they park their electron transport chains? Yeah, their electron transport chains and their ATP synthase machine are found in the plasma membrane. Now, in mitochondria and chloroplasts, the proton gradients establish a acidic compartment in the intermembrane space or the thylakoid interior, respectively. Where is that in a prokaryotic cell that is going through either process? Where does the pH get low or full of hydrogen ions? It has to be on either side of that plasma membrane. Is it the cytoplasm or is it the extracellular space? Okay. And I'm not going to answer that right now. I want you to think about it. I want it to be a question that's bothering you, okay? So that when we talk about it in class, it's very satisfying. All right. Where do you think the Krebs and Calvin cycle is happening in a prokaryotic cell, since there isn't any matrix or stroma? A neutral, both of these are sort of neutral-ish fluid-filled compartments where biochemistry can happen. In a prokaryotic cell, where do these have to happen then? Yeah, there's only one place to put it, and it's in the cytoplasm. Okay, so that's all I'm going to say about that right now. I need to move on to this last topic, which is the endosymbiotic theory, which explains the origin of mitochondria and chloroplasts and the early evolution of eukaryotic cells. So let's begin with who came up with this theory. That's right. It was Lynn Margulis. Basically, what she proposed was that these organelles that we think of as being these defining eukaryotic organelles, mitochondria and chloroplasts, actually evolved originally from free-living bacteria that had either the ability to do respiration or photosynthesis, respectively. They were engulfed by a larger cell and over evolutionary time developed a symbiotic relationship so strong that they could no longer be separated from each other. Now, what are the lines of evidence supporting this theory? Write that into the chat. That's right. Both these organelles have their own DNA. And furthermore, that DNA is a piece of circular DNA, much like a bacterial chromosome. It does not resemble a eukaryotic chromosome. They have their own ribosomes, and those ribosomes do not resemble eukaryotic ribosomes. They are 70S, not 80S. The way they reproduce looks suspiciously like binary fission, and they have double membranes, which would indicate that they may have been endocytosed. They respond in similar ways to antibiotics, to some bacteria as well. Okay, so this is in a nutshell kind of a picture depicting what happened or was thought to have happened that led to chloroplast and mitochondria evolution. And the last thing that I want to mention before I sort of open up for questions, and there's a couple other things about the Unknown ID project that I want to talk about, is just reminder and expansion on the Unit 1 test. So on Monday, we're going to do a Unit 1 review. You want to have completed your study guide for that. You want to come to class with your questions. It's going to be student-led largely, but I'm here to discuss everything that might be on your mind and to remind you how the test is going to work. But, and then on Wednesday, actually, we're going to start Unit 2. We're going to talk about growth. And we might even start growth sooner on Monday. So be prepared for that. I would prepare for growth over the weekend. What Unit 1 test is going to look like is you are going to make sure that you go to lab on Tuesday. So most of you already go to lab on Tuesday, but if you go to lab on Thursday morning, you must go to lab on Tuesday morning next week instead. Thursday morning, there's going to be 2 time slots. You can attend either one of them on Zoom. So you'll come to this Zoom link either at 10 o'clock in the morning or at 12:20 p.m. Those are the 2 lab start times, but you can choose either one. You don't have to stick to your normal lab time. And you're going to log in on Canvas. So you'll need your a picture ID, and you're going to come to the Zoom, put your name into the chat. I'll call on you. We'll do the ID check. I'll remind you of the rules of the test, and then you'll get started in Canvas. The rules of the test are that you are allowed your textbook, hard copy notes, a calculator. You are not allowed to search the internet, and you are allowed to speak to other students during or after the test about the test. You also are not allowed to click out of the Canvas testing window. So once you start the test in Canvas, you need to stay in the Canvas testing window and don't click out of it to check email or to open a Word document, and certainly not to open Google. If you have a conflict with these times, let me know. If you have other questions, we have time in the Q&A to talk about that. So one of the things I want to mention is what's due Sunday. Cells review quiz is obviously due Sunday. Your lab 3 is due. Pre-lab 4 is actually due Monday, but you want to get it done if you can. A reminder that you want to attend lab on Tuesday. And to prepare for next week, because we may actually start growth early. We might only need half of the class on Monday, like the first hour, to do our Unit 1 review. And so then we'll get a jump on growth. So it's really important that you take some time this weekend to learn about growth and watch the video lectures, even though you also have to study for the test, but don't fall behind on that. So again, the start times on Zoom will be either 10 or 12:15, and you can actually come early. I will open the Zoom at 9:30 and also at noon, so you can come early. Okay, last thing I want to talk about is the unknown ID. You've submitted, or you will have submitted check 1 for sure by the end of this week. Most of you have already submitted it, and I'm grading them as they come in. And so for part 2, you want to make sure to read the instructions and watch the video. You may have questions at that point. Before you go charging blindly ahead into part 2, if you have questions, let me know. And certainly don't start part 2 until I've graded your part 1, so that you know that you're correct. Lanisa, all students in person on Tuesday lab. Yes, that is correct. If you have a Thursday lab next week, you must come in person on Tuesday to do lab instead at whatever time you normally come to lab. If there is a problem with that, you should let me know straight away and we'll see what we can do. So unknown ID part 2, there's a video to watch. You must watch it. I can't stress this enough that you should watch it, watch the video. But you will be starting with a dichotomous key, and you want to go through it one step at a time. So you're going to ask for a test, and then make sure that you're asking for a test we actually have, using the correct name for that test, and a reminder that you want to put your name and ID number into the subject line. And you're going to use the microlabs email. And then ask me for help if you're confused by anything, but do make sure you watch the video first. One thing that I want to just mention, so I'm going to go to the course shell, and why isn't it letting me go back? That's annoying. I want to go to the course shell and share my screen with you while I just point something out. Okay. So I want to point out, many of you are in a dichotomous key that has a particular part. There's an easy part for some of you, there's a hard part for some of you. So let's have a look. So let's have a quick look at these dichotomous keys. So once you know your gram status and morphology, you're going to pick one of these 4 dichotomous keys that I've already provided. Just take a screenshot, paste it into your lab notebook. But you can see that some of them start with this decision point, the catalase question. And this is just asking, does your organism make the enzyme catalase? And some of them start with this one called oxygen requirement, and some of them start with this one. So I just want to explain how to start. These ones are the easiest. Catalase, asking if your organism has catalase, just conveniently, the way to do that is to ask for something called the catalase test. And so that's a fairly easy thing to do. But some of you start here, and you cannot ask for the oxygen requirement test because that doesn't exist. Instead, you have to figure out what test would allow you to determine the difference between a facultative anaerobe and an aerobic, an obligate aerobe. And so there is a test that can do this. We're going to talk about it in the next lab, and it's in the growth section of this course coming up. So you can preview that. It's also in the lab manual, but I'm just going to tell you that it's called the thioglycolate test. So you should go read up on that. And the fastidious test is basically you would ask for a picture of it growing on a TSA plate. Okay, question from Aminata. For the lab, are we using the average to calculate CFU per square foot hour? Yes. All right. And John, oh yes, if you're here from Saturday, thank you for asking that, John. If you're here from Saturday, you will not take the test on Thursday. You will take it on Saturday. And that's going to be actually in person on Saturday next week, not this Saturday, but next week. You will take it on Saturday of next week. In about halfway through the lab period, we're going to migrate over to the computer lab, and you'll do it sort of in person, proctored on the computer with me. Is there a time limit for the test? It is a 2-hour test. That is right. It is timed. Okay, so let me just, I'm sure you'll have lots more questions, which I'm going to get to. I just want to stop the recording. My cat is just going nuts back and forth, pacing. It's lunchtime for him. All right.